almost there are around 3,000 diseases and disorders, and most of the diseases and disorders do not have a treatment. So almost most of the diseases and disorders are unmet medical need. And if you are going to go with a target based on a discovery, right? It also has its pitfalls, and mostly it is not extremely successful. And you see most of the drugs in the market are off-target drugs, even though they started with a particular target. So when you bring in this omics into drug discovery, you are going into pathway-based drug discovery. Or you can also say function-based drug discovery. Right, so you can identify a pathway which will be involved in a disease and disorder when having just a protein and a target. Because the human body is integrated from head to toe. When anything is going to happen in the head can also happen in the energy organ. So this is an integrated approach and a holistic approach for drug discovery. And the other thing is, again, I would like to caution that these drug discovery tools are tools. And then you are not going to make a decision based on what the output is. So I was just talking to a student in the morning. So we are trying to do this within our deep training with the clinical samples, and we wanted to find out the clinical project. So he actually wanted to find the relationship between sleep and this cardiac failure, which is so common. And when I was talking to the student 3 years back, we were predicting that oxytocin would be that protein which would link brain to heart. And after doing several analysis with 3,000 clinical sample data, now the student was telling me that oxytocin is not coming out. And we also do proteins in animals, and we did metabolomics in the clinical samples, etc. So we have to go with our gut feeling, and we should exactly know what we want to do. And then we are going to essentially base our decisions on what the tools will be predict. And these tools will be predict only based on the data which you give. It's only based on the data sets. So we should use human intelligence as the interface for artificial intelligence. Anyway, these tools are extremely important. It can fasten the drug discovery process by several years. So I hope this workshop would be useful for these students, and they would learn a lot from this workshop. Thank you.
Thank you, sir. Thanks a lot for your inspiring words. Now I would like to invite Professor Ruchika ma'am, our organizing secretary, to inspire us with a few words.
A very good afternoon, everyone. And I would like to start with the welcoming all the students who have come here for the workshop. And yes, this is for the first time probably we are doing a pre-conference workshop in spirit. So this would not have been possible without the constant efforts of the students. So I would like to congratulate the student team who have put such a wonderful event, and they have been working day in, day out for this. So all the best for the event. The head sir has already given you a caution that yes, AI is important, but you need HI, that is human intelligence, as a support for the use of AI. So please, whenever you're learning any tool, so please keep that in mind and then start with it, right? And we have very good experienced the session coordinator will be taking up Professor Aprajita. So try to make the best of the event and try to learn, ask questions what you have within the time frame, so that whatever doubts you have, you can clarify them. And I wish everyone very fruitful learning and all the best to the team and for the next 2 days of the event also. Whoever has come for the all 3 days, they are welcomed and yes, all the best. Thank you very much.
Thank you, Ruchina, for your inspiring words. Now, I would like to invite our esteemed speaker, Dr. Aprajita Khan, for the inspiring words.
Am I audible? Okay. So, hello everyone. I am Aprajita Khan. I am an assistant professor in the Department of Computer Science and Engineering at IIT BHU, and I feel extremely welcomed and honored to be a part of this workshop. So, when students approached me that that you are planning this kind of workshop, it really thrilled me that this kind of initiatives are being taken part as in the course and curriculums and in the academic sessions of our BHU, and it's a real pleasure. So, but before starting, I just want to know a bit about the background that the participants have because it really helps to organize and understand what to explain, what not to explain. So what is the undergraduate or other background of the participants? Roughly, if you can, one or two participants can say. Sorry? No, that's undergraduate, but what's the discipline? For example, is that engineering? Bachelor of Pharmacy. And just another question, what kind of exposure to this programming languages do you have? Are you familiar with at least some programming language, maybe C, it could be R, it could be Python, or totally not familiar? Can I get to know? Because this is more of a hands-on session. So maybe any response? No? Okay. And laptops do you have?
Yes.
Okay. Okay, so we can start. We can try to do whatever best we can from the resources and the expertise that we already have in here. Okay. So let's start. So today's talk is on AI-driven precision medicine through integrating multi-omics. Before starting, this is a bit about myself. So I did my undergrad and I'm trained in computer science and engineering. I did my PhD from Indian Statistical Institute, followed by postdoc at Stanford University. Should I continue? Yes, fine. Should I continue? Okay. So I did my postdoc from Stanford. Prior to joining IIT BHU, I was a faculty in computer science and engineering. And these are my research areas of interest. So from a methodological perspective, I'm more of a core machine learning and statistical person who likes to work in linear algebra and optimization over, say, Euclidean and non-Euclidean spaces. But there is also this application area of interest, right? So in application area, that's where biomedical data science really fascinates me. So for example, I'm very interested in cancer genomics and cancer epidemiology, large language models, how large language models can be used to do electronic health record analysis and trying to do generate real world data real world evidence from electronic health records. The other would be computer vision and remote sensing based interest, but I'll not go into that. Okay, so let's see. So this is the age of data, right? So everything that we do, so the experiments that we perform, the smart watches that we wear, the photos that we click, they are all generating data, right? But then the question is that we are generating a huge volume of data every day. So this is you can see such big number over here. We are generating such a huge volume of data and diverse amount of heterogeneity in the data every day. But the question is, how do we make sense of the data? So it's not possible to make sense of this 320, like maybe billion, 3.28 billion of data, billion TBs of data every day. So how can we automate understanding the patterns in the data sets? So let's try to break up the buzzwords that are there. Artificial intelligence, machine learning and deep learning. So are they all same or there is some difference? And if you are in the pharmacy and other departments, so which one we can focus on, right? So artificial intelligence, so this comes from the question that we have a lot of data and we like to make sense of the data. Now humans can naturally make sense out of the data. If you show some patterns, some photos, a person can identify, okay, this is a photo of my dog and then this is the photo of my friend or something like this. So you see the natural intelligence is already built in with us, right? But when you have piles of data loaded up, then it's not possible to always do a manual look at the data and finding out the pattern. So these days also doctors, when you show the doctor maybe a CT scan report or an image, the doctor really looks into the images and the reports and tries to make a diagnosis or a set of actions that can be taken based on the report. But think about if the data piles up, and then in India we have a lot very constrained and resource crunched healthcare systems where doctors are really busy. So when the data piles up, can you use this artificial intelligence and other things to help automate reasoning from the data? So artificial intelligence would be any technique. So this is kind of the umbrella term, which is any technique that enables machines to mimic the human intelligence. But it cannot just mimic, right? It doesn't have anything. Computers are just a box of transistors. So it does not have that intelligence. So how do you? So then it comes to the machine learning. So machine learning is basically the set of computational and statistical algorithms and methods, that's where computer science come in, which are used to identify the patterns. And if you have done a basic course or maybe heard some of the terms, you have heard about terms such as maybe linear regression, logistic regression, decision trees, support vector machines. So these are the set of statistical and computational algorithms that can find the structure in the data that can enable artificial intelligence. But then our objective of achieving thing was that we would like to mimic the human intelligence. So human intelligence basically comes from where? It comes from our brain, right? And deep learning is a subset of the machine learning, which you can think of as the mathematical model of a brain. So in our brain we have these neurons which are connected. It's not that one of the neurons does all the job of remembering entire past to directly predict this is my dog from 2025 when I visited, say, US. You don't say like that. It's not one neuron. It's this interconnected network of neurons which work together in harmony to give you that intelligence. Now if you can come up with a mathematical model of the interconnected cells in our brain that is able to mimic the human intelligence better. So that's why deep learning is that subset of machine learning which uses multi-layer neural networks to directly learn from the data. So it's a subset of the machine learning that is the base of the brain. So now we come into like what are the applications? Why do we care about AI, machine learning, computer vision and all this deep learning? So let's look at some of the applications in general domain. So for example, data could be a set of Excel sheets, or it could be maybe images, it could be text, or it could also be networks. Some of the examples, for example, we have automated driving. We have cars driving through the roads, trying to identify collisions and automatically driving, right? So so the input to that model is basically, so we automatically recognize when a person is driving. How can you automate that driving skill in a machine? So that's a computer vision problem. So the input data, so I'm talking about the input data type because it's very important to understand, because in biomedical also we have very different data types. So here we are talking about that the input data, if it is an image or a video, then it comes under the computer vision aspect of machine learning, and we use computer vision models such as convolution neural networks and its variants. Now the data could also be text. So for example, this ChatGPT, all of this language models that you use, all this, so these are text models. So you can upload a text and you can ask it to summarize. Can you summarize the main findings of this research paper, and it does it. So it's a text-based model. So then these are language models where you have temporal relationships. So there are different models, different kinds of models for text-based analysis. The data can also be graphical. So graphical, for example, you have for example, in a social network, you are having, you would like some post or like some Twitters, and then you have the social networks, and then it could be graphical, and you are trying to identify the patterns within that social network graph that you have, right? So I'm trying to draw an analogy between the biomedical data sets that are there and the general data sets that are here. So can you tell me some AI machine learning application where the input data is an image? An image-based AI machine learning. Just a hunch from the background. Yes.
MRIs and X-rays.
Right. So you have the MRI, and the model tries to predict that if there is a brain tumor, where is the brain tumor, right? So this comes under computer vision aspects. Methodologically, it comes under the computer vision aspects. So this comes under image classification and image segmentation problems, right? So you have to go through the methods. This workshop is more about the genomic data. But let's just touch base to see if you can understand what kind of. So maybe an example of a textual data in biomedical applications.
Like basically the genes, like for example the conjugate which like adenine one with like the entire transcript of those following.
Right. So ATCG, those sequence of genes, that's a sequential models, and they only make sense if they code to a particular gene. They have a particular sequence of arrangements, right? And then based on that they have a certain expression. And then you can analyze those gene sequence. So definitely this is a textual data where you have temporal sequence. And then anyone can tell about this graph-based data in biomedical applications, maybe someone from that side? Graph-based data. ECG, ECG is more of the temporal data actually, but you can form a graph out of it. Yeah. But there is this gene expression networks you try to understand. So genomic data can be very easily formulated into graphs where you have genes, and you try to find out which genes are co-regulated, which genes are going up and down together, which gene is interacting with this, and you can find a graph out of it and find out that which genes are co-expressed and try to find. So we have these analogies with the general machine learning community and the biomedical data science and pharmaceutical community. Okay. So what are the applications? Now that you have understood the different genomic sources, so data sources, what are the applications? So the application is, for example, enhanced accuracy. So it's not actually, so as Sir said, it's not really replacing the human intelligence. It's assisting the human intelligence. So when you have piles of data loading up, then can AI model give a prediction which can inform the doctor to take the right decision. So these are just clinical decision support tools that can actually enhance the accuracy of prediction of diagnosis. When there is this crisis situation, then maybe the machine learning model can help. Although domain knowledge and expertise is very much important for accurate diagnostics. And then we have, for example, personalized treatment. So we'll come into that part, and there can also be, for example, patient care and resource utilization. So among different biomedical based applications, let's talk about how AI can enhance cancer research. But first let's try to understand why do we care about cancer. So you can see this is the statistics which has that how cancer incidence and death due to cancer is increasing over the years. So for example, in 2022 we had 20 million cases with almost 10 million deaths. And by 2030 there will be 24 million cases with almost 12 million deaths. So we have, and then a lot of this proportion is actually added by, although cancer is not the most highly incident disease in India, but still a lot of the burden coming from our huge population, a lot of the global cancer burden is contributed from India. So for example, this is a statistics that says that almost there are this 1.5 million new cancer cases that were diagnosed in 2025, and at least the likelihood of cancer incidence in India is almost 1 in 9 people, and breast and lung cancer are the most prominent. So we see that there is a huge cancer burden that is there in India, and we are trying to see how can we use some AI techniques to find patterns in the data extracted from patients to mitigate this huge burden that we have. So what are some computational methods? So one of the applications that is the theme of this workshop is precision medicine. What does it mean? So suppose you have in a standard framework you have a set of patients, and then if all of the patients are having, for example, let's say just say lung cancer, so you give the same solution, maybe the same drug to all the lung cancer patients, and then some of them are giving standard response, some of them who do not respond, and some of them are having adverse effects of their treatment. So personalized medicine is actually looking into the data. The data can be different types, but the data that we are focusing over here is genomic profiles, because this cancer is a genetic disease. It starts from this genomic irregularities, right? So we start looking into the genome. There are different levels of the genome. For example, you have genome, metabolome. So my biology can be a bit weak, by the way. It cannot be. It may not be exactly consistent, because we analyze it from a mathematical, computational perspective. So pardon me if some words are here and there, but I want you to get the overall idea. So we have different levels of the genome. For example, we have genome, microme, proteome. So how do you collect data from these different levels of the genome and then try to integrate the information of dysregulations at different levels to come up with a different therapy for the individual, such as so people who are having standard response, you give the traditional dose that you are giving, but people who are not responding, maybe you can increase. So the disease was lung cancer, but maybe people with stage 4 lung cancer have a higher tumor burden, they have a higher spread of the tumor across the body, and therefore the drugs needs to be maybe a bit more. There can be a combination therapy instead of, say, a single therapy, right? So you can increase. And if someone is having adversity, then you can give other alternate drugs, right? But then how do you come up with these 3 groups? To identify the 3 groups for whom you should give the standard drug, for whom you should give the increased drug, or for whom you should give an alternate drug, can that be achieved computationally? So we introduced this concept of different types of data. So we have genomic data, metabolomic data, proteomic data. I want to take an overall view of this biomedical types of data that show up in healthcare, and we call this as multimodal data. So what is a multimodal data? So a patient goes to the hospital at initial diagnosis, and the patient is following up, right? So for the first time, maybe the clinical data is recorded. So what is the age of the patient, what is the sex, what is the smoking history, and for example some demographic and other factors, clinical factors, what is the smoke exposure and other things. But then later the doctor prescribes some medicine or maybe a follow-up test and other things. All of that gets added. So those will be clinical data. And then the doctor says that why don't you get a genomic profiling done, right? Or why don't you get a CT scan done? Or why don't you get maybe an MRI done? So these are multimodal. So every time you visit, so data is spreading in terms of the modalities across the different timelines. So you have the first visit, the second visit, and every time you have different data, different types of data coming out. So that's a huge amount of data. But now how do you ingest so much data to come up with the precision medicine structure? Okay, so let's see what kind of data are there. So can you give an example of a tabular data? Data that can come in Excel sheets. So tabular data means clinical information, clinical variables that can come in Excel sheets. Why are we trying to distinguish between the data types? Because the AI model that you want to apply depends on this data. if the data is in form of an Excel sheet where it's very segregated and structured, then you try to apply some statistical machine learning. Again, there can be data which is time series. For example, this ECG signals. So ECG signals are data that are time series. So these are time varying signals. So then you try to apply some other kind of AI/ML algorithms. So then there are also text data and imaging data. And you can integrate these kind of data from different sources and do some machine learning and other methods. But today we will focus on the genomic data because the title was this multi-omics integration. So these days we do have a lot of integrative works that are trying to integrate genomics information with the imaging information, with the textual information to get a very high very condensed and like patient profile. But here we are talking about just focusing on the genomics, because within genomics also there is a lot of diversity. Okay, so let's look at the different levels of the genome. So we have, for example, the epigenome, the genome, the transcriptome, and the proteome. So now, if you have a common set of, say for example, if you have 500 patients, then you can get this. You have this next gen sequencing methods which can give the genomic sequence from the epigenome, from the genome, from the transcriptome, and from the proteome. So mathematically, we call this a multimodal learning problem. Why is it called multimodal? Multimodal means that you want to look into the patient's genomic profile, but there is not just one view of the data. There are multiple views or modalities of the data. So the same patient, so mathematically in machine learning or statistical machine learning, we try to see data in the form of a matrix. The rows are the samples and the columns are the features. And you try to apply the machine learning methods to identify some similarity or difference between the samples. So we see it as a matrix of n samples and say b features. But the point is, now you don't have just one source of the data, you have multiple sources of the data. So there you have, so suppose from gene expression you have n samples and suppose 5,000 genes. Again, from that 500 patients microRNA expressions, you have 500 samples but maybe 200 microRNAs. And then from the say proteomic expression, you have those 500 samples but their protein expression. Now the pattern that is there in the gene expression may not be exactly the same, although it's of those same 500 patients, but it may not be the exact same thing. So gene expression data will give you the idea about what dysregulations are going at the genomic level, but it will not capture that because of those genomic irregularities, what is being reflected in the proteome. So therefore, if you would like to find a precision medicine or a targeted therapy for the patients, you cannot just look into the genomic, because then you are not looking into the after effect that comes from the genomic dysregulations. So that's why in multi-omics what you have to do is, you need to integrate the information from the genomic, from the proteomic, from the epigenomic, and from microRNA and whatever other genomic modalities are there, and this term itself is called multi-omics. So these are called multi-omics. So we have multi-modalities. Each of the modality is a omic modality: transcriptomic, genomic, proteomic, epigenomic, and therefore we call multi-omic analysis. And so that would maybe one who in a like biomedical data science background will call, and from the CS background we call it as a multi-view machine learning, where instead of one data source you have multiple data sources. So for example, in remote sensing you have the same part of the earth being captured by multiple satellites. So the algorithms that has been developed are not necessarily distinct or biomedical data science problems and computer science problems. They try to treat the problem mathematically, saying that there are independent views of the data which have some consistency because they are from the same samples, but they are not exactly the same. Because if they are exactly the same, there is no point in capturing the multi-omic. You can just have the genomic and find out the gene expression. But sometimes you cannot just design a drug based on the gene expression changes. You need to also see if the protein expressions are consistent or not, right? So that's why you need to integrate the information. So I'm trying to motivate that the modalities are different, they are heterogeneous, and we cannot treat them distinctly. We need to integrate them. So that's why the title was Integrative Multi-omics. Okay? And then once you integrate them, then you can try to find out that what are the common or consistent dysregulations across the different genomic levels, and you can find the therapy that targets that. That will be a more holistic therapy compared to just looking at the protein expression and suggesting a drug, and based on the gene expression you suggest another drug, based on the changes in some microRNA expression you suggest another drug. That wouldn't give a global comprehensive view of the dysregulations that are happening in the cancer. So you computationally integrate the data, then find out the differences among those n or 500 patients, and then you design the drug for those different subsets of patients. Okay? So that is the overall. But the objective is how do we achieve it. So before we can achieve all these computational things, you need to first find the data. So you need to start about where the data is. Okay? So anyone knows where multi-omics data is present in internet? Anyone heard about the Cancer Genome Atlas? Right. So we are talking just in case of oncology, cancer research. Therefore, I will be introducing this repository, which is called the Cancer Genome Atlas, TCGA. And the data of this, so this was an initiative by NIH to capture the multimodal omics data for cancer patients in US for certain time period, and they created a huge database for which most of the majority of the patients has these multiple modalities. For example, the same set of breast cancer patients will have their genomic, gene expression, protein expression, microRNA expression, and other things available, and you can computationally integrate that data to find out some patterns. So this one is called the Cancer Genome Atlas. But then this is available through two portals. The first portal is the UCSC Xena browser, and the second one is the MLOmics. Now those who are with your laptops, can you take out your laptops and find and do this search, UCSC Xena browser, and the other search would be the MLOmics Python package. So I want you to really look into the data. This is not a slide discussion. Right? Did you get this? UCSC Xena browser. So let me show you how to browse. People got this? UCSC Xena browser, है ना? UCSC, or you can just write TCGA Xena, HCML. You guys can check if they are able to see. Those who have laptops, some of it is like not all are not required, but just show them like this is Xena browser. Are you able to open this Xena browser? Laptops anymore at the back. UCSC Xena browser. Are you able to see? Okay. So let's browse data sets. People have UCSC Xena browser loaded. So let's really try to see the how does the multimodal data look like? Okay. So you go to data sets. Do you say like this?
those who have laptops, you can sit together if possible, then others will also be able to see. Although I'll do it over here. Okay? So let's go into maybe, so these are, you see, so these are the different types of data sets from different initiatives. So we have this lung adenocarcinoma, lung squamous cell carcinoma, melanoma, mesothelioma, ovarian cancer, pancreatic cancer. So we have all these types of cancer data sets over here. So let's try to log in one of the, I'll just click on this lung adenocarcinoma. So did you go UCSC Xena browser, data sets and lung adenocarcinoma? Are you able to see? So what it means is that this is a cohort of lung adenocarcinoma patients, and what is the total? So for example, they have copy number variation. You understand what is copy number? Genomic copy number? Huh? So this is like whether how many copies of the gene are being are there. Right? So you see you have this copy number variation. Then you have, for example, this gene expression. So let's try to click on this one. So gene level. So if you click on one of those, it actually shows you that there are 503 samples, and for all of those samples, you have the gene expression. So for example, this one is the gene, this is how gene expression data looks like. So these are the patient IDs. So they are from that cohort, and these are the ensemble IDs of the genes. So for example, you have TP53 and other genes. These are different genes and the genomic expression level. So for example, if this is 6, it means that this gene is overexpressed compared to this gene's level in this patient. So basically, in this patient, this gene is having an expression level of 2, but this is a 3-fold more expression of this gene. And then maybe that means something. Do you get it? So this is the gene expression. So this is one of the omic modalities. Other multi-omics. Okay? So when you download and if you want to really do AI and machine learning, you start with this matrix. It looks very fancy, gene expression, epigenomic and other things, but essentially you start with the data matrix of a same number of 503 samples. Okay? Then you have another modality, let's say this is DNA methylation. So you see this is the different omic modality, and these are the different regions of the DNA. And then you see the expression, the values are much different. In the other table, we had values from 2 to 6, but here the values are very small. So what I am trying to say is that the data may not be of the same range. So each of these data might have very heterogeneous expressions. So what is 6 over there is not equivalent to a 6 over here, because here in this modality 6 does not occur. So there is this heterogeneity. You cannot just combine and find out that okay, that gene is expressed, that means this patient also this DNA is 6. So they are very heterogeneous. So in terms of machine learning, it poses a lot of challenge. So for example, again you can go to, for example, this one, this is another gene expression. So I just want to check if you are able to get to the data sets. Able to look at the different modalities. Okay, if it makes sense, at least the terms. Okay. You may not have a very comprehensive understanding of the genome, but at least if you have to start, if you have to do AI ML, you have to start with the data. You cannot skip the data part. Right? You cannot skip the data part. You have to look and try to understand what the data means. Whether there is a pattern in the data or not, will ML model will do. But you have to first find the source of data. If you want to do multi-omics, you have to first look into this Python packages or Xena browser. Xena browser is something which is official. So officially this is the data repository. But the next one is the GitHub repository, which is a like Python where you can directly code and download the data. You don't have to go through this selecting the data set and downloading. Okay? So it starts with finding the sources of the data and how to access it. So this is the paper which is in Nature, where they put together our Python library to access this data. As you can see that these data sets are very high dimensional. That they have a lot of size. So you see there are 60,000. There are 60,000 genes for 500 samples. Do you get it? This means that there are 60,000 genes for 500 samples. If you try to download this, many times your download and other things may be paused and you may not be able to find any pattern because the data is too huge. The whole genome profile is too huge. So that's why you have this kind of Python packages. Okay. So I can understand many of you may not have a Python background, but trust me, if you want to survive it, you better learn Python. And the earlier the possible, better. Okay. Don't skip that Python. You have to come to this computational. Research is really moving towards this AI driven. So it's better you get your hands on the Python and try to understand how to do. These days you have a lot of like Copilot and other things which helps to do the Python code. You don't need to do it and learn from scratch, but you have to start learning Python. Okay? So for example, here this is how to get the data set directly from Python. So it has, you see, it has the cleaned data set from all these types of cancer. You see, esophageal, head and neck, kidney cancer and lung adenocarcinoma. One muscle carcinoma. So I'll switch back to the slide. So I'll switch back to the slide. So essentially, in order to access multi-omics data, in order to access multi-omics data, you have two. One is the browser where you can select the data you want to download and you can download the Excel sheet and you can look into it. Another would be the MLOmics database where it has a very good documentation. If you are somewhat familiar with Python, you can install the Python package and just select the data set. It will be a clean and pre-processed data set that will be downloaded. Okay? So this is just whatever we say. So this is a browser-based workflow which does not involve any coding. However, like for example, if you would like to access it through this Xena browser, you might have to do some coding. But thanks to the students who helped me build this tutorial, they have already downloaded the data set and cleaned it up so that we can directly start the downstream analysis from the data. Okay. So basically this old TCGA, the Cancer Genome Atlas, this is the cleaned version, but it has almost, let me see, so it has more than, I think more than 10,000 patients or something. So the clean data set has more than 8,000 patients for 32 types of cancer, for 4 genomic modalities, and it supports at least 20 different tasks including classification, clustering and missing value imputation, and it also has different types of pre-processing genome. So first you try to browse through Xena browser and then you try to use the GitHub. Once you are familiar with the Xena browser, you try to access the data set not from the Xena browser because it will download multiple files with multiple names and other things. You try to use this MLOmics Python package to download. Okay? So during the coding will show. Or maybe what we can do is, Pravinya, we can start simultaneously. So I was thinking that I will do some part of this slides and then we'll go to the code, but we can do it back and forth. So what I need to do is I need to share the Google Drive link to all of them. Can you share that? Okay. So she will share a Google Drive link. I will also open up that link and then we'll try to fire up this Google Colab and try to do the computational part of modeling. Okay? So is it shared with the
Can you send me? Oh, I think I already have that link only you are sending, right? Okay, so Google Drive.
I am really missing the laptop. Why didn't you come? You young students, why didn't you bring laptops? So did you get access link for the spirit workshop folder? Participants? लिंक मिला आपको? हाँ हाँ लिंक नहीं जाता अच्छा अच्छा तो इसका मतलब ओपन एप्लीकेशन I can do that. This is the portrait. Now this is the portrait. What's up? What the name? What happened? Oh. I need to sign up. I can sign up. Google Drive folder. Did you get link? Or do I need to do this QR code thing? Oh, you have to enter your email address. No, this is not the QR code. नहीं मिला अभी तक गूगल ड्राइव लिंक नहीं मिला मिल गया सबको मिल गया गूगल ड्राइव should I stop bothering myself with QR codes and stuff हाँ ठीक है okay fine so are you here those with laptops are you here student workshop folder spirit workshop folder हाँ हो गया ठीक है now go to the data ठीक है go to the data folder ठीक है so there are many files here if you try to download I think it wouldn't take much time but we don't need to download all the files in case the network and other things don't work for safety we have all of these files what you need is this first file and what you need is this last file ठीक है so in your machines you download this first file multiromance.itynb and the lgjsurvival.com did you reach through this folder yes ma'am ठीक है पहला फाइल को अपने कंप्यूटर में डाउनलोड कीजिए आखिरी वाला ठीक है जस्ट डाउनलोड so see I have started downloading है ना want you to do is, in your machine, have a folder which has just these two files. Okay? This much you can do. Go to the Google Drive, download the first. I think the students can help. So what are download कर लिया? हाँ? ओके कर सकते हैं? खुला है खुला? So after downloading, what I have done is, I have uploaded it to my drive so that I can launch Collab. Okay? And I am doing all of this so that I don't edit the master file. Basically. So what I want in your computer screen is to load this Python notebook in your Google Colab. Okay? We need to ensure at least that much is done before we can go to other steps. So maybe the TLs can help. Loading. So in your own Google Drives, you have to upload, and then the Google Colab should be loaded. What are the Google Colab, right? ठीक है कोलैब पहले कौन कौन यूज किया है नॉट इट्स फाइन ठीक है सो अभी तक हाउ मच डिड वी डू सो वी डिड द डेटा लोडिंग राइट सो Xena browser में तो we saw ना how to go to that Xena browser and explore and find out where the data is. Now this is the second way, the programming based way to the Python package. ठीक है? So these are called so basically this Colab is a IDE, integrated development environment, which is basically a code editor, but it's kind of a notebook. So basically you can also write other things and you can also run the code, and the output of the code can be displayed in the same file. So this is called a Google Colab. So this is totally online. We are working with online things so that in case you don't have some packages installed in the library, in your own machines, it will still work in Google Colab. Okay? So this is how the interface looks like. You basically have this kind of text cells which are called markdown, which is fine, but you have the code cells. You can run the code directly over here, and the output that is generated will also be shown here. Okay? So how to run? There are shortcuts, but I would suggest you that you can just try to click the first button, or maybe it's trying to mount. Na? Okay? First we can. So अगर फर्स्ट ऐसे बहुत कंपलसरी स्टेप नहीं है, इसको स्किप कर सकते हैं. ये थोड़ा छोटा फॉन्ट हो रहा है. So if you are clicking on that play button to the first cell, Did it run? ठीक है, it will take some time. So see, once it runs successfully, it gives this tick, है ना? रन कर पा रहे हैं? हाँ बताओ. हाँ, ओके, proceed. हाँ. गूगल कोलैब आप पहले नहीं करते हो तो वो एक बार वार्निंग देता है. It's fine. Just proceed. To do this Python machine learning, if this interests you, कि हाँ ये सब सीखना है, AI for precision medicine करना है, these are the fundamental things that you should start to learn. The Python libraries, ठीक है, the infamous Python libraries, pandas. This is called data input output. Then you have NumPy for numerical Python. ये जो लाइन मैंने लिखा है ना, pip install pandas, NumPy, matplotlib, Clone, scikit-learn. ठीक है? So these are very important libraries. Even if you don't understand what is going on, not clearly, you should try to go home and try to learn these libraries. Pandas, NumPy, matplotlib. ये एक दिन में नहीं होगा, पर तो शुरू करना है ना? ठीक है? So let's try to learn. So once your notebook is loaded, then the first step is to install the packages. ठीक है? So that whatever functions you call in the code, it can call the implementation of those functions from those packages. ठीक है? So you run this one, है ना? So for my machine, it may run a bit early, I'm not sure. So you see, it's running. ठीक है, तो ये टिक आ जाता है तो मतलब आपकी जो कोलर है उसमें वो डेटा आ गया है. Python packages are installed. Are you getting the tick? So getting a tick over here is very important for the next cells. ठीक है? Okay, so once you have installed the packages, you call the packages, you import the packages, saying that in the next part of the code, I will be using specific functions that are defined in that packages. So that's why in Python, if you want to import packages, the code is import. तो मैंने पहले क्या किया? I loaded the notebook in Color, then I installed the necessary packages, and see now I am importing those packages for use in my code. ठीक है? This things may be not trivial, but the students can help you understand. So you see, you try to run this cell. If this one is running, check. So it's basically importing the packages. So now you can use all the functions that are defined over here in this package. रन हो गया यहाँ तक? ठीक है. ठीक है. अभी ये देखते हैं. So हम लोगों को so you see, so basically I am giving this website where the data set is present. It's called MLOmics. So this is the way to. The first way was from Xena browser. The second way is from the MLOmics. So let's see. You see, I have given this link where the MLOmics package is there. ठीक है? And then I am importing its data to my main data sets. I am loading whatever is there in this website HTTPS Hugging Face. उसको मैं अभी अपने मशीन में ला रही हूँ. ठीक है? So you run this. ठीक है? Click on this. And you open up this folder जो साइड में है. You see, this main data set will be created as a folder here. ये शिवम इन लोगों को ये folder दिखा देना. So when you are running this, here you see it's importing the data. Do you see that blinking? So it's basically receiving the objects. So now you don't need to browse through the Xena browser and download manually. Through this Python package, you are able to import all the data, all those data sets that was there in the Xena browser. You see वो blink हो रहा है ना? ये थोड़ा time लगेगा, but it will import the data in your Google Colab, and then you can use it. We will show using one of the data sets, but it will import all the data sets. ठीक है? This road हो गया? हाँ? अच्छा हाँ ठीक है वो थोड़ा टाइम लगता है। आप सब लोग कर रहे हैं ना तो उसको और थोड़ा टाइम लगता है। हाँ मतलब एटलीस्ट कुछ शॉर्टकट बना लेते हैं। That's why you have to package no one cycle package is not bad. So that you don't have to do it through that Zena browser or anything. हाँ select कर सकते हो और क्या चलेगा then you can just rest. So you can study using the lower grade learner, but you can then do the experiment on the learner. तो ये Python तो सिंपल है ना? ये तो आप साइड में तो ये वाला एक्सपेरिमेंट बना लें और वहाँ पे एक पेज है So you see, once this one is run, can you click on the main data set in the right hand side and see what data sets are imported? You get it? We will show with only one example, but click on that main data set. Side में main data set आ गया ना? See, so many data sets will show up. Classification. Now you check over here, Zena browser. Zena browser कहाँ था? यहाँ पर. You see this? TCGA, BRCA and other things. See, TCGA, BRCA. Now look into your Google Colab. You see TCGA, BRCA. You get it? So whatever was there in the Zena browser that we had to click and download and do, now they are already imported over here. So here all this data set, breast cancer, colon cancer, glioblastoma multiforme, lower grade glioma, ovarian cancer, and cancer. Then within clustering data sets you have all this kidney cancer, lung adenocarcinoma. So you see all the data sets are here. See, within the data sets you have all the folders. So let's click on one of, just browse and see how it looks. So you see, if you click on the top, acha, I think it just downloads there. So do you see this? Are you able to see this? You see, for the lung adenocarcinoma, it has those 1, 2, 3, 4, 5 modalities. Do you see? Copy number variation, methylation, microRNA, messenger RNA. जो UCSC Zena browser में देखा था ना, वही वाला now it's in Colab. So I want you to just see because later you can experiment. In this folder structure. Okay, so this Python package not only gives you one data set, but it gives you at least 10 to 12 multi-omics data sets to start exploring. Okay, should we move on? अच्छा और maybe we can close the session now and then have the break is right. At least people देखिए इतना तब आपका सेल रन हो जाना चाहिए है ना post टी ब्रेक सेशन के लिए ठीक है if this cell is running it's fine for now you are fine. Looks like no tea for 15 minutes. डेटा सेट आपको दिख रहा है अपने मशीन में? हाँ? This is G is over here chemistry. OB is the short form. Then click on this folder top. Click on top. So over here the copy number variation, methylation, labels, and passenger RNA, mRNA and microRNA. This is the multi-modal. You note this again later. Are you able to see what data set? So you have this data set, right? Okay, so let's go data set of let's say one code करना code set. It is very easy. Oh अच्छा अच्छा ये हाँ ये मेरा में हो जाता है I think it's I'm trying to find आप इससे बाहर चले गए ठीक है
How is it going?
Top row filter, copy number variation, methylation, label, mRNA is gene expression, miRNA is microRNA. You can also click on this and like see what it looks like. Maybe it will click or will show it in the later part of the ठीक है, so can I close this window here, browse, right? ठीक है, so we close this so that we can focus on this one. So this one will do the job of importing the data set to your Google Colab. So the Xena browser data through the MLOmics package has been now imported in your Google Colab, and you can choose the data set to work with. ठीक है? Okay. So now we want to load the data set, right? So you see, we start with. So suppose I am just choosing TCGA, GS, LGG. LGG is lower grade glioma. It's a brain tumor, right? So I am choosing. So you see, I am setting the path. If you want to experiment with an other known data set, this is where you change the path and give the disease of your choice. Suppose you want to work on some other cancer, say kidney cancer. Here you have to see what is the code for kidney cancer in TCGA. And that path name you have to change. ठीक है? So this will give you the folder where the multi-omics data CSV files are stored. ठीक है? So then what do? You see what is PD? PD is for pandas. Pandas is a library in Python for data input output. So I am using the pandas to do read CSV. This is basically reading a CSV file. CSV is comma separated file जो Excel में लोड हो जाता है, ठीक है? So it's reading the CSV. What do I read? You see, I'm reading mRNA, which is gene expression. miRNA is microRNA expression. CNV is copy number variation, and methyl is DNA methylation. meaning of this output? Print command is printing what? So it's printing the shape. So overall it's a genomic and everything, but for computationally it's a data matrix, we said, right? We are starting and working with matrices. The matrix has dimension what? Matrix is n cross n. N rows and n columns, right? So I am doing mRNA dot shape. This will give you the number of rows and columns in the matrix. So do you see something? So it says that, so you see, this is 5,000 for mRNA, 200 for miRNA, CNV 5,000 and MET 5,000. Cross 247. Can anyone help me understand what is the meaning of this? इसका क्या मतलब हो सकता है? mRNA is gene expression, है ना? So if it is 5,000 cross 247, you see 247 is common. Do you see? Yes. And in the slide where we did, where is our slide? So in our slide, if you remember, over here we were doing sorry. So in this we had these n samples, common n samples. So for all of those 247 you have protein expression, gene expression, copy number variation and DNA methylation. तो अभी जो स्लाइड में आपको दिख रहा है वही आपको कोलाब में दिख रहा है, right? Do you agree? How many samples do you have in your data set? 247. So that means in the lower grade glioma there are 247 patients for whom you have the gene expression and 4 modalities or multiple omics. ठीक है? Okay. So people are clear up to this cell. Should we move forward? Yes. ठीक है. So then we are loading the survival data. इसको अभी रन मत कीजिए, ठीक है? इसको अभी छोड़ देते हैं क्योंकि ये एरर देगा, ठीक है? This will give error. ये pin code, right? This will give error because this one read.csv इसमें नहीं है ना. Okay, so sorry, they have fixed it. Very good, thank you. So we can run and this will also execute. So I'm basically loading the survival data. I will say what it means. Can you just run? Okay? So up till now my data loading is done. Next we'll visualize and find out patterns. But इतना तक आपको रन हो जाना चाहिए, ठीक है? This cell till this one. I think this is cell number 6. No, not 6. Working. What kind of experience? Okay. So, let's go. Wild guess. Okay. So once you have loaded the data set, let's run the next cell and see how the data looks. So अभी तक मैंने CSV files import तो कर लिया, but I did not check how it looks. So let's try to run this. Run this cell df.head. So essentially, you are giving the path to the CSV file that has the data. You are using the pandas read CSV function to read the data in the CSV file, and you are loading it to the variable df. ठीक है? So let's try to see how survival data looks like. Anyone can give a wild guess on what this columns mean? What is the last column? What do you think is the last column?
Patient ID.
Patient ID, correct. What is this second last column?
Either it's a 0 or a 1, like indicating some feature is present or not.
Feature is present or not? Okay. Let's take another guess. So you can change this, है ना? ये जो head command है ना, it shows the first 30 rows of the data. You can change it to say 50 or 80 to see more rows in the data. What it means is 0 means that the patient is alive as per TCGS record. 1 means the patient has died. And this is the time in days how much the patient was followed up. So this patient maybe left the study after 1 day. Maybe he switched to a different hospital. These are number of times in the days. So how long the patient was followed, and until the last follow-up of the patient in the hospital, if the person was alive or dead. So you are trying to find out that the longer the time, that means the better survival. So this is survival data. We will try to find the survival difference by statistical modeling later, but you have to understand what the data means. So survival data has a patient ID. For each patient you have two things. The first one is a binary status of whether the person is alive or dead until the last follow-up. If it is 0 means the person is alive. If it is 1 means the event has occurred. In this case the event is death. The event can also be, for example, cancer recurrence, or it could also be cancer metastasis. Different data has different modeling types. If you are trying to understand when cancer progresses, then the event should be 1 when the person moves from an early stage cancer to an advanced stage cancer. But here we are trying to follow up that which person survives longer. Therefore, we have one column which is a binary column, 0 and 1. 0 meaning the patient is alive and 1 meaning the person has died. And then the time is how long the patient was followed up in the hospital before the person dropped out of the study, maybe for some external reason or maybe the person died. Okay? We'll see what is the utility of this kind of data. Okay? So you see, the .head command is basically showing the first few rows of each data. Okay? So can you run this command, mRNA.head? And can someone tell me what it means?
Is it some normalized score, like कोई particular scale पे ले रखा है ताकि सब data set को एक ही?
No, this is not normalization. This is just, head means it's just showing the first few rows. It's not applying any scaling, nothing. We will have to normalize, but this is not. Okay? So up until now what have we done? We have loaded the data set. Let's see. So what are my variables? mRNA is the variable that has gene expression. miRNA has microRNA expression. CNV has copy number variation, and meth has DNA methylation. Then what I do? The first important command is dot shape, which gives the number of rows and the number of columns. And the next important is dot head, which shows what is there in the first few rows. Right? So for example, if you see the first few rows of the survival, it looks something like this. This is how your gene expression looks like. RNA or gene expression looks like. Okay? So basically, this is saying that this is the patient ID, and for the DPP6 gene, this is down-regulated, minus negative expression, whereas this one is up-regulated, positive. Okay? But here we are not working with one modality, not omics. We have multi-omics. So therefore we have another called miRNA.head, which is showing the expression of the microRNAs. small segments of the gene. Sometimes they are the coding sequence of the gene and their expression also matters. So then you are trying to look at the copy number variation. How many copies of the gene are there? Right? And the fourth is DNA methylation. So if you are not working with one matrix, working with one matrix is really easy. That's like classical machine learning. You go to Kaggle, you download a data set, it's one data set, it has n samples and some features, and you directly run. What makes biomedical problems challenging is that there is not just one view of cancer. The cancer dysregulation can be in genome, in the microRNA, it can be in the DNA methylation and several sections. Therefore, you have to work with 4 big matrices. You saw the size, right? 247 rows and 5,000 columns. Okay? Am I making sense? Or should I wrap up the workshop and everybody is happy? Okay. Okay, so what should I do? Do I go for a break or what? Okay. Okay. So then what we are doing is, you can just run this, it's called transpose. So essentially I want the patient in the rows. Earlier the patients were in the columns and the gene expression genes were in the rows. So I just swapped it for notational convenience. Okay? So I've just done a transpose. Matrix transpose you understand, right? So I'm doing matrix transpose. Okay, so let's go to the slide. Okay, so we'll go back and forth. So essentially, इतना तक हम लोगों ने कल कर लिया है ना, एनालिटिक्स we have already imported. ठीक है? Then you have the data set, the first part of the analysis. This is the primary analysis, and then you have the secondary analysis. Primary analysis is machine learning, and secondary analysis is biostatistics. We'll do both, hopefully. So the first part is load the data set. Load हम लोगों ने किया है, ठीक है? Next is harmonize, reduce and cluster. So there are 4 steps. First you load the data set, then you harmonize. Since we are using a package, they are already harmonized. But if you had done from UCSC, then they would have different types of samples. One would have 237, one would have say 325, one would have 200. So in that case, you have to find out the common sample. Let's go over the steps. So the primary analysis in multiomics is load, harmonize, reduce and cluster. So we'll go through that. So can you relate to this slide? You have seen this, right? We just showed. That not head commands. So we have this RNA sequence matrix, then we have methylation, here we have the clinical data, copy number variation and protein expression. We have it in the Python, you see? The 4 modalities we have it here. 1, 2, 3, 4. That exactly same thing I am showing in the slide. Okay? So loading done. Next is harmonize. Since we have used the package, they are already harmonized. There are 247. But sometimes in the individual, when you saw in the UCSC Zena, you see this N was the number of samples. So copy number has 468, but gene expression has 447. So these are not harmonized because this was the raw data set. We use the Python package, so they are already 247. You don't have to do the harmonized step. But sometimes you want to, for research purpose, not work with a pre-processed data set. You would like to find the patterns starting from raw data set. In that case, you need to download data set from here and run the harmonization. Harmonization is what? Basically, you have different modalities, different omic modalities, and they have different number of samples. You try to just find out the common set of samples. Okay? So that you can do the multi-omics analysis. Because if you just do gene expression separately, then you will not be able to find out that the clusters or the patient groups that were formed based on gene expression, is it the same homogeneous group that is formed in microRNA? So there will be the difference. In that case, you will have to give a different therapy based on gene expression, and you have to give a different therapy based on microRNA expression. So what do? You take the common set of samples. That's the harmonized step. You take the common set of samples and only use those samples. It's called complete analysis. But there is also incomplete analysis where you can take those, random different set of samples. We will do the simple part, which is complete analysis. So you see here we have patients 1 to 5 and some other patients. These are some other IDs. So it's only 5 patients who are common. Similarly 247 patients for LTG. ठीक है? तो इसका क्या करना है? Basically you have to take the intersection, right? Coding में कैसे करेंगे? In Python it's very easy. Do you see this line? Common samples. So I'm basically, sorry. Am I not sharing screen? Why is it not done? वो आ जाए कैसे? Do you see? So what am I doing over here? I'm basically harmonizing. So I have loaded the data set and now I have a common set of 247 patients and there are 4 modalities. ठीक है? Fine? Should we can break at next?
So now we will be taking a short recess. We will be having a 15-minute break and refreshments will be served outside. I request everyone to please leave their laptops here as a lot of us have provided our own personal laptops and we need to take inventory. Right? So I request you please understand and please leave your bags here as well. We will have this entire area sealed off so you do not need to worry about security. And refreshments will be provided outside. This is only a 15-minute break. We ask for your understanding in this time. Thank you.
it fine? Is it not working? Is it showing the same screen? So now is it showing which screen? The code screen.
Yes, ma'am.
It's showing the code screen, right?
Yes, ma'am.
So basically I have to switch the windows also in Google Meet or not required? Oh no, we can then switch to code screen as the main screen. So अभी code screen दिख रहा है.
Yes, ma'am.
Then mostly this is the major screen. First half like okay. Okay, so should we start? I know people are exhausted and this is not a particularly very interesting topic to do on a Good Friday, although the name is Good Friday, but I can see that it's already coming back because of me, but I think we can still make it a moderate Friday, right? Okay, so to make it a moderate Friday, we have some code cells to go through, okay? So overall we'll do some ideas. जो लोग you are trying in the laptop, it should run and you should have an overall picture of the story, how it flowed, okay? Even though you don't know the code specific things, you should have an overall flow. So can someone summarize what we did so far?
First of all, we imported all the relevant packages that were, the pandas, Skype and all of that. Then we started on, we went on to visualize how the data was, and not like visualize, but see how the actually data was organized into rows and columns. And that's it, and then we we take the exercise from the data in order to take the common values out and then start out.
Right. Okay. So let's recall what is the goal of today's this workshop session. That we want to understand a very basic application of precision medicine. What is meant by precision medicine? So we went to that slide, if you remember. In this slide we talked about this precision medicine slide we did, right? Which means that basically for diseases, advanced diseases like cancer, you should not give the same drug to all the patients. Or if you give the same drug to all the patients, someone might have good response, someone might have negative response, and someone may have no response, and someone might have some adverse response. For example, they get some maybe like comorbidities and other things which you want to avoid. Okay? So how to avoid that? That we want to do targeted therapy. That is called precision medicine. The medicine is not generalized medicine. So if you see in doctors, we have this general medicine, right? Doctor specialty. But in oncology, we have the specialized. So precision medicine. We are trying to do that how to find out precision medicine application of something from the data. Doctors can of course do it. Doctors can look into the profile, but that is manual. It takes a doctor's time, it takes a lot of involvement. We are saying that if we had the patients and we had their genomic information, how can we find out whom to give different drugs? ये हमारा overall question था. So the question is that we want to design a targeted therapy application. Whom to give different types of therapies. But then whom to give we are trying to answer from the data. So basically, given a set of 247 samples means 247 lung cancer patients, the question is that do you give the same drug to all the 247 patients or do you give different drugs to different patients such that they have good response? So basically, you have this group and it's about patient grouping. Do you get the overall idea? Target medicine means finding out the groups of patients who have similar profile so that they can be given the same drug. Now, the whole patient if we see, then there is heterogeneity. But within this subgroups, maybe there is more homogeneous. The genome profile is more similar. So we are trying to find out from their genome expression, microRNA expression, protein expression, and DNA methylation. We are basically trying. Sorry, what happened? Okay, so it will take a moment to fire up because it's out of charge, but we can discuss the problem statement. So basically, given the 247 patients, we would like to find out that what are the 2 or 3 different or 4 different groups of patients such that their genomic, transcriptomic and microRNA profiles are similar. Based on that, you can give separate drugs to the 3 subsets of groups of patients. Instead of giving the same drug to all the 247, what are the different groups and what drugs can you give to those groups? Okay? So abhi we have loaded the data set up until now, right? And then we have also harmonized the data set that they all have same sort of samples and they have different the gene expression, the copy number and the variation. Other things. उतना तक मेरा data load हो गया, ठीक है? अभी मुझे क्या करना है? I have to find out the groups in the data set. So in machine learning, this problem of identifying groups in the data set is called clustering. Have you heard that term before? Clustering. So machine learning has different problems. One is classification, another is clustering. If you are trying to find out the groups in the data set, it's more of a clustering problem. So essentially what we will do, we will take a data set which has many features. It has 5,000 genes, 200 microRNA, 200 protein and many things. And then we find out different groups in those matrices, right? तो उसके लिए क्या करना होगा? जैसे I just see whether it loads up. So once you have loaded the data set, the next step is to normalize. Why you need to normalize? You need to normalize because in some modalities there could be that the gene expression has very high values, but the microRNA you saw the value is between 0 to 1. But in gene expression it was 10, 15, 20. So then it's not meaningful to combine, for example, like a value that is within 10 to 15 to a value which is between -1 to 1. Okay? So now it's working.
प्रोजेक्टर का क्या हुआ?
But the point is, if you have have this 15,000, you don't really see that whether there are groups or not. How do you find out groups in this big matrix? You need to visualize, right? So if you have to visualize, then basically what you have to do is, you see, this one is called, so I'll come over here. So the next thing that we do in machine learning terms or AI, it's called dimensionality reduction. Okay, these terms they might not seem familiar to you, but if you study AI level, all of these terms you will come to understand. So first is data harmonization, then normalization, then dimensionality reduction. So dimensionality reduction is basically a technique which takes a large amount of data of a higher dimension. For example, there are 20,000 features and it tries to bring it tries to project it into a two-dimensional or a three-dimensional space so that you can visualize and see if there are some patterns. If there are 15,000 features, you don't know if there is some group. ये मैट्रिक्स देखने के लग रहा है कि इसमें कुछ ग्रुप्स है पेशेंट्स का. It doesn't look like this, right? See, the data set raw looks like this. You are not able to understand whether there is some patterns or not. So what do? You use this as a linear algebra mathematical problem and you try to reduce the rank of the matrix. This is linear algebra, you don't need to go into this. But basically, it tries to take 15,000 columns and come to a summarized 2 columns. ठीक है? 2 columns में अगर आ जाएगा तो then I can plot it in the x-axis and the y-axis. And I can try to find out that whether there is any grouping or not. If there is no grouping, then there is no need to give precision medicine. That means it's just a homogeneous group. But if there are different groups, then based on those groups you will give different medicine to different patients. So what do? You basically, you see, ये सब code है, but what I want to explain is that here you see, after I have started with the big 15,000 matrix, I pass it to an algorithm called principal component analysis. ठीक है? So this is the code for that. This is the sklearn, which is a package in Python which has this PCA, which is a Python package to reduce the data. When you have large volume of data and you are not able to make sense of the data, you would like to project that large data into a two-dimensional space like a page, so that you can make the plots on the page and try to find out that what are the groups of patients in that data set. So you see, I was not able to make any sense of the matrix, but once I reduce the dimension, I can see that they look like this. So do you see some groups in the data set? देख के लग रहा है कि मतलब ऐसा तो नहीं कि पूरा एकदम यूनिफॉर्मली गोल करके है. So there are some groups. So at least you can say that the patients corresponding to whom these dots are there, they have a similar gene, microRNA, protein profile. Why? Because I had concatenated those data sets. Do you remember? I merged the gene expression with protein with microRNA and then I reduced the dimension. So that means those two dimensions, this is called principal component 1 and principal component 2. These two dimensions has all the information of those 15,000 features, among which 5,000 were gene, 5,000 was copy number variation, 5,000 was DNA mutation, and 200 were microRNA. Now I have summarized all those information into 2 plots, x-axis and y-axis. Once you are plotting, you can actually see that okay, they are all blue points, but looks like there is some grouping where these are similar and these are not similar to each other. At least you can find that, right, from this plot. So then what you do something called K-means clustering. ये notebook को आपको फिर से देखना होगा, ठीक है? But the steps I am trying to make you understand. So then you do a clustering, which is very easy to call. So basically it's calling a function, which I am calling with 3. So then it comes out that the program is giving that these patients are of a certain type. These that were close by each other looks like a different group of person, and this looks to be a different group of person. ठीक है? Just based on those 2 dimension of plots. Whether they are actually biologically different or not, we have to do some analysis. But at least the computational way, from those so many big matrices, you are able to find out 3 groups of patients. You get it? उसमें steps क्या था? I first loaded the data set, then I harmonized the data set to have a common set of samples, then I merged the data set after normalization. After merging, there were a lot of features. There was gene, microRNA, DNA methylation, and I was not able to make any sense of that data. So I passed it to a mathematical function called PCA. It will be very much used if you do computational parts. If you pass it to a PCA, it's a linear algebraic formulation, mathematical formulation, which basically squeezes the information in large matrices to smaller matrices. So it's basically a matrix transformation. 11, 12 में पढ़ा था ना? Matrix transformation, या फिर like वो solving a set of linear equations and other things. So it's a matrix transformation. So you are transforming a matrix of 50,000 columns to a matrix of 2 columns. Once you have the 2 columns, then you can run a clustering algorithm, which is basically giving you 3 groups. Okay? So this is basically the end of part 1 of the analysis, which is my primary analysis. So it's load, harmonize, reduce, and cluster. This is your primary analysis that you should do. Even if you are not working with multi-omics, if you are just working on protein expression, or if you are working with gene expression, you will usually find out that you have 100 patients, and when you do the gene profile, they have 20,000 genes. So just leave out that harmonized part, which is for the multi-omics, but you should do load, reduce, cluster for your own multi-omics or individual omics, and you will still get the same cluster, but they are just based on say gene expression. People who had used gene expression data. Someone in the audience was interested in protein expression, so they are protein expression based cluster. They are not capturing the genetic dysregulations, but from the proteins that are generated from the genes, from their expression they are trying to find out that which proteins are dysregulated or which subset of patients has same set of proteins dysregulated, so that you can give them a therapy for it. Okay? So this is part A, which is the primary analysis. I'll quickly wrap up with the second part, which is very important. If it was a computer science participant, I would have probably skipped the second part, but the second part is a bit more important for a pharma-based background. It's called the secondary analysis. The secondary analysis is looking at that the clusters that you have got from the machine learning output, are they biologically relevant or not, which is important. You can have because these are only interplay of matrix, the code will anyway run even if there is no cluster. If it is not cancer, if it is just fever, and even if there are no groups, the Bayes clustering can give you some groups, which is not biologically meaningful, because it's just an algorithm. It works on some input and generates some output. Output is biologically valid or not comes from the domain expertise. So don't skip the second part, I am saying. So if you are designing a study, you have to think about AI-based study. You have to think about the primary outcome, which will be the output of some AI model. And then you have to talk about the secondary outcome, which will use as input the output of the AI model, and based on that find out some clinical meaning. That's what sir was saying, that AI is just a tool. You have to give some human intelligence to find that. So you have to do the secondary analysis, which is more biostatistics. It's not machine learning, but it's statistics to try to find out that okay, is this pattern significant or not? Is this pattern reproducible or not? If I do the study in Kanpur and I do the study, do I see the same differences in the patients or not? So that is the secondary analysis. Okay. So in the secondary analysis, what do you do? So basically, from the primary analysis, as you can see over here, you are getting the 3 clusters of patients or subgroups, right? The secondary analysis takes this as input. You get? So it takes those cluster labels as input for the second stage analysis, trying to find out what is the difference between the clinically difference between the clusters that are learned via algorithm. These are not clusters that are found by a doctor. So essentially what we are doing, we first do differential gene expression analysis. So differential gene expression analysis means that between the two groups of patients, let's say the green cluster of patients and the red cluster of patients, what are those specific genes which are, so if they are similar expressed in the both the red group and the green group, that means that is not a target that you can, that gene is not a target. You cannot use them to distinguish and have a different like efficacy. So you have to find out that what are those certain genes which are only say overexpressed or differentially expressed in only say the green cluster. Then you can find out a drug that can try to reduce the expression of those genes. But the first part is to find the groups of green, red and blue and find what are the genes that are differentially expressed in one of the clusters. Okay? So this is a differential gene expression analysis. In the interest of time, I'll skip over this coding part which you can look at because this is there. What it gives as output is basically, what is it? So you see, here we are showing, for example, it's showing a list of say 10 genes. So it's showing that these are the 10 genes that are differentially expressed between the cluster 0 and cluster 1. So that means these genes are specific to the cluster 0. That means the green cluster. So if you have to design something for the green cluster of patients, you should basically try to understand what these genes are responsible for and try to predict or come up with design a drug design a drug that targets these genes, because these genes are specific to one of the subtypes, and the AI part is to give you the subtypes. You are not like manually finding that okay, these patients have same symptoms. They might have same symptoms, but they might have a different genomic profile. So that's why you use AI to find the different and find the specific genes corresponding to each cluster. Once you have the cluster, so this is this one. So this is the post-clustering analysis or the secondary analysis. The first part would be you identify cluster-specific differential gene. Once you find out the cluster-specific differential gene, you basically try to do this gene ontology, pathway and survival analysis. Pathway was what sir was saying. Trying to find out what genetic pathways are different. So you find out what are the specific genes and what are those biological pathways that the gene is responsible. For example, maybe some genes are responsible for the apoptosis, which is killing of the cell. So maybe that gene is not working. So then you have to find out that whether that gene is differentially expressed in a cluster and that is responsible for maybe the apoptosis pathway. So then you can come up with a drug that targets that gene. So computationally, as you can see in this notebook, if you run, it's called so this one is for gene ontology analysis. So if you do the gene enrichment, that means you are trying to find out that what are those pathways where these gene is enriched. They have a role to play. Okay. So if you run this, what you are getting is that, see, these are the pathways. So for example, the first most important gene in one of the clusters is the negative regulation of tumor necrosis factor. So that's one of the gene ontology. So that is one of the gene that is kind of influencing this pathway. So if you are trying to target a drug for those set of patients, you should try to design a drug that targets these pathways. So the second step in the secondary analysis is to find out the pathways that are like modulated or enriched in the differentially expressed genes. Okay. That's the second analysis. And then finally, in the second analysis, you have the third part, which is called the survival analysis. So survival data, you remember we loaded it, right? Survival data has basically two fields. One is the binary status, whether the patient is alive or dead, and what is the time the person was followed up. So you have now three clusters, three groups of patients. So what I am trying to do over here is that this one is called a Kaplan-Meier plot. So this is a survival plot. I pass that survival data and the cluster information from the stage 1 to this function. It basically gives the survival profile of the different groups of patients. What it means is that, so basically you see this cluster 0 and cluster 1. Cluster 0 is orange, cluster 1 is green, and cluster 2 is that blue. So, can you try to tell me which cluster of patients has a good survival versus a bad survival from this plot? So, the y-axis is the survival probability. Just try to understand. Also, this is the last cell, so you should be like, you can try to understand this one. So, tell me, this one is the time, the colors of the different clusters, and then this one is the survival likelihood. Okay? So, which cluster of patients do you think has the worst survival among the three?
Red one. Red.
The?
Red one.
The orange one. Right? Why?
Because when the time is increasing, only the red line is there.
The survival deteriorates. So this is for example at 0.5. So, after what point the person reaches the 50% survival chance? So you see, these people in the orange cluster are reaching it at much earlier, around 2000 days. So maybe 3 years. Or no, not 3 years, but 6 years, right? So around 6 years. So if you are diagnosed with a lower glioma, which was our disease, you are likely, the most likely that your survival maximum is of 6 years if you are belonging to, let's say, this cluster 0 of these patients. They all have lower grade glioma, do you get it? They are all LGG patients, 247 patients. But among them also I see that the survival is very different between different subsets. So once you find out these survival differences, you can actually try to target a different drug or more effective drug for this high risk patient groups versus this one which has a lower risk. So for example, its survival probability reduces to less than 50% after 3000 days. So 1000 more days. So maybe 3 more years. Right? So we are trying to understand. But you understand that we started this with just the gene data, which had nothing. It was just a matrix over here. See, we installed all these packages and other things, and we started with this data, which was not meaningful, right? But then he tried to do what? He tried to organize and harmonize the data, normalize the data, reduce the dimension. They had many features. You tried to reduce the dimension and tried to find out the clusters. What are the groups? Once you have the groups, then you are doing all those analysis, 3 analysis. First, differential gene expression. To find out which genes are specific to a cluster that are overexpressed or underexpressed compared to others. For those differentially expressed genes, you do gene ontology and pathway analysis to find out what pathways they play an important role in. And then you look at the survival to see that whether the clusters that you have got, they do have some clinical differences in their survival or not. If they are, that means the clusters that you have got from an AI model is also clinically meaningful. It's not necessary that the AI model will always give you the clinically meaningful clusters, right? So essentially, if you are trying to do, always do 2 parts of the analysis. So I will summarize by saying that basically multi-omics, it reveals a lot of human biology, and it's basically a scalable pipeline, and then TCGA is the starting data set. But this is how it starts with. So you first load the data, then you harmonize and normalize because the data is heterogeneous across gene expression, DNA methylation. So you harmonize and normalize. Harmonize means you take the common set of samples. Normalize means you adjust for the difference in values. The third step would be, because now you have combined gene, DNA, methyl, it has increased a lot in the dimension and it does not make sense. So you reduce the dimension using an algorithm called, let's say, PCA, Principal Component Analysis. You will get to know if you just dive a bit. It's a very important algorithm. So now you reduce the dimension to 2 to visualize if there is groups or not. If there is groups, you computationally find out. The algorithm is called K-means, very simple. Once you find out the groups, then you find out that what are the differentially expressed genes and pathways in those patients groups. And finally, you see what is the survival difference. So step 5 and 6 are basically secondary analysis. Step 1 to 4 is primary analysis. So this part is driven by AI and machine learning. This part is biostatistics, trying to clinically validate the outcomes of the AI model. You get it? So you have to do both the parts of the study. You get, so this can also be done manually. So a human can find the cluster, which was done traditionally, and you can try to find out what are the pathways and design a drug, or you can use this AI pipeline using Python and other tools. You get it? So I'll conclude. So I'm very thankful to the entire Spirit team, especially Ruchi ma'am, along with the students who have been super helpful in setting up, especially Pravinya and Chirayu, right? A wonderful efforts. They have given a wonderful effort, and the organizing team, Riya and Anushka, and all others who are here. I would also like to thank my computer science engineering research scholars, Anshika, Shivam and Rahat, for putting their efforts today in trying to help with the hands-on session. And thank you so much for attending this workshop.